This recording is part of the material that we typically deliver in person as part of our Applied Bioinformatics programs. It is included in our Introduction to Data Science workshops, which are designed to provide a high-level overview of essential bioinformatics data skills. In these workshops, we cover key topics such as concepts of code and data, project management and organisation, and as you'll find out in this session, robust research methods. These week workshops aim to equip you with foundational skills to approach bioinformatics research and data analysis confidently and effectively. So in this session, we're going to cover robust research and include focus on examples around sanity checks and quality control. So before we start, I'd ask you to take a few moments, so you might want to pause this recording if needed, and consider what you think of when you hear the words robust research. So on this slide, we can see a word cloud that highlights the key concepts and values that underpin robust research. Terms like scientific integrity, reproducibility, transparency. These all remind us of the foundational principles that guide reliable and ethical scientific inquiry. So robust research is often characterised by practices that prioritise precision, validity, accuracy. It's not just about conducting experiments, but ensuring that the results are reliable and that they will stand up to scrutiny. For instance, you can see words appear like phrases like peer review and verification. And so we think about external validation of our findings. You might see words like methodology, rigour and statistical power. And so these highlight some of the technical aspects that you might think about before you start your research to ensure it's robust. So how we design our studies and handle data to ensure that the conclusions that we can draw are statistically sound. But ultimately, robust research also demands accountability. So you can also see reflected in here with the terms like ethics, integrity, quality assurance. So robust analysis refers to the statistical methods and approaches designed to produce reliable and accurate results. Now, real-world data is rarely perfect. While we might hope for some clean and, I think if you're working with R, we might call tidy data sets and standardised data sets, the reality often falls short. These next couple of slides focus on some of the strategies for navigating the challenges of what we describe messy data. First of all, we're always going to aim for tidy data, but you must be prepared to encounter the opposite. Even when collaborating with the same team or individual, you are likely to face inconsistencies in data formats and structures. You've got to keep this in mind. It's not a reflection of anyone's efforts. It's simply just the nature of the beast, the nature of real-world data collection. In some teams, they will implement standard operating procedures. You might implement standard operating procedures so you can minimise the variability in your data format, but it's not always the case. So processing and quality control is going to be an indispensable set of steps in your workflow. Tasks like identifying missing data, missing values, resolving inconsistencies, standardising formats, they're just critical aspects of your work, and it helps make your data both reliable and actionable. To streamline your work, I recommend, and something that I do in my daily practice, is to establish clear data harmonisation standards. So this might include adopting consistent nomenclature for variables and file names, using common genome builds for when you're working with certain data sets, genomic data sets, or establishing core reference data sets that you're going to align your data to. This can reduce discrepancies, and it also promotes interoperability of data sets. So I'll be able to use the same data set in multiple downstream pipelines. Remember, when you first see data that's messy, it can be quite overwhelming. But it's part of the growth of the bioinformatician, and it's an opportunity to develop and refine your data management and analytic skills. If you can implement structured processes and start adhering to established standards, you can quickly get into the process of transforming this kind of disorder into clarity. I recommend embracing this challenge. It will be part of your day-to-day life, having to deal with difficult data sets, and it's going to be an essential part of your research journey. So, working with real data, as I said before, does mean encountering errors. It's not a question of if, but when. So first, you've got to acknowledge that every data set could be prone to mistakes. These errors can arise from a variety of sources, including some data entry issues, some system glitches, some inconsistencies during processing of the data. But recognising this reality is going to be a first step in mitigating its impact. Never just assume the data's perfect. So you've got to start by asking yourself, can you check for errors? So can you implement some validation processes using automated tools or manual inspection to identify any inaccuracies or inconsistencies in the data? Can you correct those errors? And actually importantly, should you correct the errors? Well, some errors can just be easily fixed by writing a bit of code to replace one value with another. Others might reveal some deeper systemic problems. For example, you might be correcting some errors that could be hiding broader issues in the data collection and processing workflows. And I'm going to give you an example from some work that I did on a genomic study. A large international data set that was could be downloaded by researchers around the world. So I had access to both the genotype and the phenotype data. And whilst reviewing the data set, one of the things I do is I run a simple process of comparing the reported sex from the phenotype file with the sex that I can determine by looking at the individual's genotype. So I look at the X and Y chromosome and I run it through a pipeline, and it can tell me what the reported sex is and the genotype sex is. And what I noticed, because I compare the two, is I noticed there was a mismatch between the genotype assigned sex and the recorded phenotype data. Further investigation, and I found that the discrepancy seemed to arise after a specific data collection date. So it wasn't throughout the whole data set. We got to a certain point in the data collection and this error occurred where the discrepancies seemed to happen every other sample. So this pointed out to me the potential issue in the data processing. So upon I informed the research group, and when they did further investigation, their study management team discovered effectively what was a copy-paste error in the phenotype data. They'd missed a row out. And so that one individual being missed, or the numbers, the IDs being assigned to the wrong person, caused a shift and a misalignment in all the records after that date. So I could have easily corrected the sex code, going like, oh, that sex code's wrong, I've got the genotype sex code, I'll replace it with this new data. But that would have hidden the broader issue that all the other phenotype data that was coming my way in that file was misaligned. It would have gone unnoticed, and it would have impacted all the downstream analysis. Effectively, I would have been just randomly shuffling the data and not necessarily doing the analysis that I wanted to do, asking the questions that I wanted to ask. So thankfully, the root issue was identified, it was resolved, and ensured the integrity of the study. The study management team was also able to effectively recall the original data and release an updated phenotype file to other researchers who would access this data around the world, who there would have been many, who some may have spotted this, some may not have, but now at least they know that their analysis, they can redo their analysis knowing that the phenotype and the genotype data are aligned, and actually the phenotype data is aligned to the right person. So the final thing that I've kind of flagged up here is that I was able to, and actually the group over in at the study management group for this project that I mentioned, were able to correct the data, and on their side, they would have been able to keep detailed records of where the errors occurred. And this is going to be essential to ensure that you have transparency and that other processes are reproducible. So I always say to students, try not to edit the file manually. Try and write some code to do any updates to a file. So when you start off with your data, any edits that you do, you can track them. And the next person who runs that analysis can see what's happened, what went from that original data set to the next step of the analysis. And this documentation, or either through code or through actual physical documents describing what you've done, it maintains trust. If I see that in a data set, I trust the data a bit more because people have shown me what they've done. And it adds integrity to the work and allows others to follow your methodology with confidence. If you systematically address these errors, you will enhance the reliability of your studies. Remember, data imperfections are inevitable, but how you manage those imperfections is down to you and can set the foundation for you having robust and trustworthy research. So in the next set of slides, what I'm going to do, I'm going to delve into some examples to kind of show you really and illustrate how mistakes infiltrate even the most trusted data sources and some of the cause of that, and show that actually sometimes it's not your fault. So these examples aim to demonstrate that no data set effectively is immune to errors. So let's take a step back. One of my personal frustrations when I started working with gene-based data, and actually with most bioinformatics data, is that when I came to want to submit this to a journal, there was a reliance by the journals, and there still is, on using software such as Microsoft Excel. They always like to see the spreadsheet. They want to see an Excel file that contains your data and your results. And so Excel is widely used in offices and laboratories. I don't have a problem with the software per se. It's pervasive in most workspaces, but it does have a well-documented quirk that poses significant challenges when we talk about scientific research, specifically on genes. So by default, if anyone would open up Excel now and type in gene names like SEPT1, SEPT1, or DEC1, or JUN1, Excel will interpret those, if you don't switch off the defaults, as dates or floating number points, and automatically convert them, and that completely alters the scientific meaning and can render a data set inaccurate. So if you've suddenly got 01-September as opposed to Sept1, when you try and do some data merges with that, you've suddenly got a misalignment with the nomenclature, and actually it's renamed a gene incorrectly. So this was something we were aware of, and actually this published paper on the left of gene name errors are widespread in scientific literature. This came out in 2016, published in Genome Biology, and they highlighted the scale of the issue. So the authors found that approximately 20% of papers contained some Excel-derived supplementary files that included errors of this nature. So it's quite a large number of papers that they looked at had this error. And so 20% of these papers had some unintentional impact on the integrity of their scientific data. So what did we do about this? Now, did we all stop using Excel? Did the journals stop asking for Excel? No, they didn't. But the community got around and said, we need to rename the genes. So these genes were renamed to avoid any of these automatic conversions. So for instance, Sept1 is now being renamed to Septin. It preserves its scientific meaning, and this workaround demonstrates a critical point. Sometimes it's necessary to adapt some of the scientific standards to align with the limitations of common tools that we all use, rather than expecting Microsoft to tweak their software for our needs. So in this second slide, I want you to have a look and see if you can spot any errors in this one. So this is a scatter plot, and the data is showing the maternal and paternal age at the birth of a child, and it's plotted in months. At first glance, you can see, hopefully, and I'll see if I can get this on the screen here, there's a reasonable correlation. Moms and dads tend to be similar ages. And so that's good to see. It wasn't completely scattered. It follows what we expect to see. However, as researchers, it's our responsibility to critically evaluate this kind of plot and scrutinise it for some potential issues. Now, I've used this example because you'll have some understanding of what maternal age and paternal age looks like. And so let's take a closer look and think critically about what stands out as unusual or incorrect. And we kind of think about things like outliers, range consistency and symmetry. So I'm going to let you thinking of those terms, just look at that data, pause if need be, and try and think of some data points there that you would question. Okay, so let's discuss some of the possible anomalies. So first of all, let's think about outliers. What you want to do, you want to look for some of the data points that deviate significantly from the main cluster. So in this example, let's look at the lower end of the plot. There's a data point here, around this point here, and that suggests that father was approximately 6 to 7 years old. And that raises immediate questions about plausibility. Could this be due to data entry or some misunderstanding during the data process? That could have been something that was inputted as months instead of years, or the other vice versa. Now, if we saw some of these inconsistencies over here, you'll go, okay, we've got on this side, we've got a mother who is likely to be quite young here. And then we might also see some disparities between the age of the father and the age of the mother. And those questions, they might look like they're outliers, but they might be biologically plausible. They might be real data, but you still might question some of these. So that comes to that second point of range consistencies. Are all the ages biologically reasonable? So for instance, if we notice maternal paternal ages far exceeding natural reproductivity limits, that might indicate error. It might not. If you see individuals that have large age discrepancies, that might also be true. You may have a 15-year-old mother and a 65-year-old father. Biologically, that's plausible. It seems to be a bit of an outlier in society, but it could exist. And again, I mentioned before, we look at the symmetry between the distribution between the maternal and the paternal ages. We expect to see some form of correlation. If we see a massive skew, or if we see a complete random distribution of data, we might ask some questions about some systemic biases, inconsistencies, or data collection formatting. So these errors might result from various sources: data entry, mislabeling, software errors, conversion issues, lots of different points. Or they might actually be real data, but it's good to just double check when we see things that are just outside the norm. And trying to identify and addressing these problems at this stage, before we go off and do some downstream analysis, might be crucial in maintaining integrity before processing with those downstream analyses. Let's have a look at the next example. So, for those of you who've worked in a lab, you might spot this. This is a slide that gives you an example of something called an SGS page gel. And this is a technique in molecular biology that looks for protein separation and analysis. So this method is widely trusted. It heavily depends on the skills of the people doing the experiment and their execution. And as we can hopefully see from this gel, it demonstrates several issues that undermine its reliability. So if I produce in the lab and I say, let's do some follow-up work on this, I would probably sit down with my colleagues, with a student or my supervisor, and go, I need to redo this. So there's a couple of things that we see on this gel. Uneven bands. Some of the bands appear smeared or irregular. This might be something to do with how you loaded the gel. if some of the samples degraded or the gel wasn't set properly. And that makes it really difficult to interpret your results. There's lots of background noise, there's high level background staining, some of the lanes are obscured, and again, insufficient wash steps, some of the technical aspects around using the gels that's meant you've got a poor gel here. You've got some distorted lanes, and it might be due to underloading, overloading of the gel, so that's putting too much sample in there. Points of technical error in sample preparation, inaccurate pipetting, uneven electrical current, lots of different things. But the take-home message, if you look at this gel, it would raise some flags. Hence it comes from the SDS page Hall of Shame, and you can go to look at these various people who have submitted their gels that they're less than impressed about. So as a researcher, it'd be critical to identify these problems, and this doesn't pass the site test. So we'd troubleshoot this gel, and we might have to go, that's it, we need to restart this experiment. So we'd have to optimise our technical ability to load the gel and verify our reagents hadn't gone off and refine our gel preparation. We'd redo this experiment. And we'd realise that we need to repeat the work because it doesn't meet the standards that we'd expect. Now, we're talking about bioinformatics, and what if we didn't look at this gel, or we weren't able to look at the data points on this gel? We didn't see this experiment happening. It went through an automatic process. You see these green lines here, and the green lines captured intensity data, plugged it into some software that ran some analysis, extracted some information, and said, yes, the experiment's worked. It's completed, as you can see by this little image at the bottom. We've analysed this gel, we've got some data, it's completed, and you're sat there with some data points that you're going to happily walk off and do some analysis on. So when working with this type of data you've had automated tools process the intensity, it might streamline the analysis, but it carries the risk of what we call silent errors. Subtle mistakes, not so subtle in this case, that might not be immediately apparent to the end user, but will definitely compromise the validity of the findings. If you looked at this manually, you would have thrown it in the bin, started again. But if you just put it through the automatic pipeline, you'd have data that you'd then have to go, what does the data tell me to whether I need to keep this or throw it away? It might be the data you're quite happy with to plug into some downstream analysis, and it tells you that gene A and gene B are enriched in this experiment. So what we do in this case, when we can't look at all the gel, can't look at all the images, we try and incorporate some quality assurance and quality control into our processes. So there's a couple of key points to remember when we think about this. An output does not equal accuracy. So just because a tool generates an output doesn't mean that the process is error-free. It's your job to put things in place to be more confident that it's not error-free. So automatic systems will produce flawed results from flawed data input. So be vigilant. You might want to review log files. Log files that come with data analysis, they try to capture some metrics that tell you the quality of your data and some assumptions of the data distributions that you might want to work with. A lot of log files will have those sanity checks incorporated in there. You might want to do some post-analysis sanity checks, so ensure the results make sense. Does it align with some of the biological or experimental expectations? For example, I did an experiment once where I looked at protein-DNA interaction, and I ran this through a mold-it-off machine to tell me what the proteins were. And what I expected from that experiment was to see one protein pop up lots, because that's the antibody that I used to extract that specific protein. So the bulk of my findings should have been this protein A. Protein A didn't exist. So it told me that actually the antibody wasn't binding to the protein that I expected it to bind to. So anything else that I found, I didn't really trust. So I started again on that experiment, because I needed to, there's some things that we will expect from our data. Now, this next example, we're going to start looking at plotting data and a real experimentation where sometimes you can't look at all the data. And this is from something called looking at automated genotype calling. And I'm going to orientate you around these plots. So these scatter plots represent signal intensity. And the colours there are pseudo colours at the moment, so don't really, the software doesn't align the colour yet. But imagine this was what you can see if you looked at this in black and white. For each of these is 3 clusters. And this red cluster that we see up here, this represents the intensity of the A allele. And so in each of these dots is an individual, and these red dot individuals are individuals in that cluster that we believe for this marker RS2910190 carry the red allele or the A allele twice. So they're homozygous for the A allele. This cluster down here would be, this blue cluster would be homozygous for the blue allele, and this green cluster here are going to be individuals that have one copy of the red and one copy of the blue. And so these are what we call heterozygous individuals. And what you can see, just looking at these plots, 3 nice clusters. You might also see black clusters that fall down here, and that would be an experiment that has failed, an individual that we didn't have genotype data on. So this is a lovely example, well separated, and we'd be quite happy if we saw this, that the genotype calls that we were doing were relatively accurate. Clear, tight clusters suggest good quality data. And as I said, not all data is good quality. This is some real data from an experiment, and let's have a look at these scatter plots. These plots highlight several potential data quality issues. Scattered or overlapping data points, so you can see on this one we've got this black line here where these 2 clusters aren't well separated, so the software couldn't tell whether they were part of this AA or the heterozygous band, so it just called them missing. A lot of missing samples down the bottom here. In this section here, we can see a well-defined cluster, but the software has decided that it doesn't like this intensity, so it's called all these individuals missing, these black crosses at the bottom here. So what we're seeing is maybe some poor DNA sample, errors in allele intensity measurement, assay insufficiency, some noise in the processing. So in this bottom one here, it doesn't have the right coordinates to be able to call the AA alleles. So we see cluster shape and spread, outliers, unusual groupings, and this ultimately leads to some errors in these data. If the errors are completely random, it doesn't have that much impact. But if the errors are specific to one cluster or specific to one arm of the experiment, then you are going to be introducing bias into your analysis. This non-random error is a real problem. So, as I mentioned before, effective analysis depends on robust quality control measures. And this might include visual inspection. And we've done that here. We've looked at these, and we know what a good cluster plot looks like, and we can see some bad cluster plots here. And that might question how we trust the data. So, in an ideal data set, we'd expect the clusters nice and distinct, well defined, non-overlapping, but here we do have these non-random missingness. So what's the solution? Should we manually review all the data plots? And sometimes if we have a data set that we're really interested in, we really want to look at one marker, but it's not meeting some of our criteria, we might go back and plot these up. But what if I told you this might involve plotting 2 million markers and reviewing those? And I did an experiment where we first were looking at this. We wrote a paper on the impact of this kind of experiment. Myself and colleagues, I plotted out a couple of thousand of these and got people to sit there and tell me what they thought of, 2 or 3,000 of these plots. And it was a difficult task to look at lots and lots of different plots, and difficult to make some of the calls between plots that may or may not be, that look worse than they actually are. It's not feasible, it's completely impractical to do that on 1 million plots. So what do we do instead? Well, we try and look at other ways to pick up these errors and these silent errors. And so when it's impractical to manually plot all different variables. What we can do is we can look at alternative metrics that might flag poor quality control. And in this case, of a genotype assay, we might look at the missingness at a specific marker. So if a marker had a very high rate of missingness of data, we might say that carries too much error, I don't trust the data, and we'll drop that marker. We might look at the proportions of the AA, AB, BB, so there's homozygotes and heterozygotes within a single genotyping assay, and that will give us some knowledge of something called Hardy-Weinberg equilibrium. So there's some knowledge that we have about the expectations of the proportions in each of those groups, and so though if it deviates from those proportions, that again may signal some errors in the data. In addition to looking at the individual SNP, we might look at the individual DNA sample. So if somebody submits their DNA and 20% of it doesn't give us reproducible results, or reliable results, or any results at all, so we've got 20% missing, that would raise to me concern about the quality of the data that we do have. So we might go, can we have another DNA sample from that individual and drop that individual from the analysis? And the final thing we might look at, like I said, looking at proportions, we might look at the proportions across an individual of how many samples are going to be heterozygote. And that tends to follow a certain pattern depending on the ancestry or the population that individual comes from. And if we see too little heterozygosity, so of those ABs, or that might indicate poor DNA, or if we see far too much heterozygosity, that might indicate that somebody's mixed two DNA samples together. And so again, that gives us other measures that can say, do I trust this data? So let's go back to this plot that we see here. This is a single experiment looking at the missingness of each SNP, and what we've got here, we've got 1.45 million SNPs. And I've made a decision at the 2% threshold here. I'm not going to keep any of these experiments that have more than 2% missing, and that's 278,000. Actually, it's a little bit more because this has missingness greater than 0.5%, so it would be a little bit more than 278,000. When we actually clean those samples out, we now have a data set, and this is what we'd carry on to do our work on with this bottom panel. So this is before quality control, after quality control. And now I'm going to show you why doing these little things, we've lost a lot of data there, but we're hoping that the nature of the data that we're using, we're not losing information from our experiment. So now I'm going to take you back to an example that I've been using. It's from Twitter from September 2019. And this is a notable case study concerning a gene called CCR5 and the challenge that we would have around real-world genetic data that we share. And you see on the left, this scientist has written, "The one thing that all scientists fear the most is to find out that a major result they have published was based on erroneous data. This is an event that will affect you for the rest of your scientific career." So they're very upset about, they've had a paper effectively retracted. And I'll give you some background on that paper. So CCR5, that encodes the receptor that is critical for some immune cell migration to sites of infection. And what was found, there was a mutation, a delta 32 mutation, which is a deletion of 32 base pairs on CCR5 on that gene, and it results in a non-functional receptor. Homozygous individuals with 2 copies of the mutation exhibit a resistance to HIV infection due to lack of functional receptor. So if you don't have this, if you have 2 copies of this mutation, you have immunity to HIV. So obviously real world impacts here. There was a controversial case where a group of scientists edited the embryos using a process called CRISPR of some babies to give them this mutation, this double mutation, to make them immune to HIV. Lots of ethical questions about the use of this approach in this scenario, but that's not for this topic here. If you want, it's worth reading around some of the stories about gene editing, especially embryonic gene editing. And so these researchers, the researcher that we can see on Twitter here, Ryan Nielsen, they analysed a study called the UK Biobank to investigate what was the potential effects of having a homozygous delta 32 deletion. So did it have any other effects? And their findings, that was published in Nature Medicine, very high impact journal, indicated potential links between the mutation and mortality risk. So there was lots of, real world implications of doing this. So consequences of editing. So would you reduce HIV but increase other diseases? However, when people looked at the data, they found some significant analytical issues in their study. Issues that could have been avoided by appropriate QC. And ultimately, the authors retracted the paper. And that was a significant moment. It's great to see scientific rigour and scientific accountability happening. And so this paper was not left for others to believe the data was true. So let's have a look at the data that they worked with. So we've seen these scatter plots before, slightly different colour coding here, but what I'll emphasise, this is that scatter plot of the genotypes, and what we're looking at here is this grey stuff. These are all the missingness. One of the things that's important to make for this, so this is analysis of a marker called RS62625034 that captures this delta 32 mutation. Let's break it down. So the missingness rate is 3.4%. I've told you before, typically in my QC I would have dropped that sample out because there's a lot of missingness going on here, so I don't really trust the data. So it suggests there's some potential issues with the assay or data collection. And actually, if you look at the plot here, you can see where the missingness is. There's lots of missingness there. There seems to be some bizarre clustering that goes on with this sample that compromises the reliability. The second metric which I mentioned is looking at the proportions of the AA, AB, BB, the homozygous and heterozygous. That's something called Hardy-Weinberg equilibrium. When they looked at that based on the observed allele frequency, what they'd expect to see with what they actually saw, they saw a massive significant skew away from the expected proportions. And the p-value for this is 2.06 times 10 to the minus 53. And all you need to know is that's infinitesimally small. Really unlikely for this to happen by chance. Okay? So, when we do our QC, I would generally, and most people should, exclude these because of their QC. They would have excluded them because of the missingness, and they would have definitely excluded based on the Hardy-Weinberg. But this might, as the authors might argue, this may have been real data. But so there's another thing that we can kind of leverage just as a sanity check. And there's this thing called a linkage disequilibrium. Some markers that are close to one another on the genome tend to be inherited together, and so one can work as a proxy for the other. And this is what this slide is. So this is additional supporting data, and this is a story provided by the marker RS113010081. And this marker is in very strong linkage disequilibrium. That means it's co-inherited together with the marker on the previous page. So these data sets should be very, very similar. And what you can see from this is that when you look at this plot, only shows 0.08% missingness, and the Hardy-Weinberg deviations aren't significant, so it's as you'd expect by chance. So something that's supposed to be the same, and actually if you just see passing the eye test on this one, if you look at it, you can see these clusters are more well defined, less missingness there, more trustworthy data. And actually when you analyse this sample using the research question that the authors did, it found nothing. So it suggests that something that is good quality data that should match what they that should also be a proxy for this delta 32 mutation found nothing. And so that should have, when you ran your analysis, should have set up warning flags to go, I don't trust that data, we're going to fix it with a marker with better quality data. And when they did that, they would have found a null, and they're reporting to whichever journal, maybe, to Nature Medicine, maybe to another journal, would say, when we looked at the impact of this, we found no impact. Now maybe that's not as exciting as a sexier paper, but it's a real paper. It tells us about the real data. Finally, the final example, you've seen a lot of plots now. I'm going to talk about how I use reference data and why you might use reference data to help inform your experiments. So these plots here, and let's focus on the one on the left with the yellow and the blue. This compares allele frequencies between a reference sample and a test sample. So the y-axis represents a reference sample, which in this case is the European Population 1,000 Genomes Project, and on the x-axis that represents a study of individuals from a white British population. So similar ancestries, allele frequencies should be the same. And what you can see on this, I've colour coded this to make it nice and easy, is the blue points are what I would say is reasonable correlation between the two data points where the allele frequencies tend to align and they're correlated. And we might carry these on for downstream analysis. So what about these yellow points? So they represent deviation, some majorly significant. If we kind of look at here, the allele frequency is expected to be close to 100% and it's actually 0%. So that suggests that the allele we're looking at may be the wrong allele. So say if we look, the A allele might be at a 98% and the B allele might be at 2%, but somewhere along the line we've got those flipped. We're coding the wrong one. And so these discrepancies happen. And they happen quite often. And the main difference is often between the sources of the genotyping array that you've used, even from the same company. Their version 1 of their platform that genotypes 1 million markers might be different from their version 2 because they've identified errors, they've identified type errors. What's clear worth pointing out here is all these yellow dots are quite significant. These blue dots are very, very compacted. There's many tens, hundreds of thousands of points under here. What we're seeing here is actually very few markers that are different. It just looks quite dramatic. And so going back to the point of you might get these errors occurring between two different data sets. What I tend to do is I go, if I don't know which one's right, I'll drop both of them. So I'm not going to flip the data to make sure it aligns to the 1,000 genomes. I'll just go, questionable data, I don't know who's got it right, I'll remove that, and I'll use that phenomenon that I showed you in the previous slide, that linkage disequilibrium, to hopefully capture the variation that I want to capture in my experiment. And so I've aligned to a reference to effectively identify differences between arrays. So I've given you a fair few examples there. So as a sign-off, I want to ask you, when you consider what you've learned in this set of slides, ask yourself how you approach your data, your experiments and your tools. And I always say, can you trust them? Try and not blindly trust everything. So effectively, trust no one. Verify everything. Learn to plot. When a lot of the examples I've shown you here is how visualization really helps us understand what our data looks like. Perform those sanity checks. Validate with assumptions. Compare your data with expectations and be consistently critical of all parts of your data analysis process. So, the goals of robust data analysis is to provide a framework for trustworthy and reliable results. At its core, robust analysis ensures that we can confidentially interpret our analysis and rely on them for decision making. Limit errors, identify errors, review your outputs, and document your errors, report them. That allows transparency. So thank you for your attention. And please note that this session is usually delivered in person as part of a broader interactive session. It typically includes group discussions and collaborative activities where you and your peers will engage with material on screen, share your thoughts and provide your perspectives. We emphasise active engagement in the programme, and the students' participation and contributions are key to making the sessions meaningful, insightful and enjoyable. So hopefully that's given you a taster of what we do. We look forward to hearing from you and hopefully welcoming some of you to our programme in the future.